it going, everyone? It's Chow here, and today we are going to talk a little bit about cell fate, so let's get started. Okay, so what exactly is cell fate? Well, you have a type of cell, a specific type of cell, that will ultimately potentially become another specific type of cell. And so this differentiation into another type of cell is often a function of differential gene expression. I want to think about it, all of the cells have the same DNA, so if you're going to make the cells slightly different, you're going to need to use differential gene expression or morphogenesis, which is necessary for the fact. So cell fate is what a cell is going to ultimately become, or what a line of cells is ultimately going to become. And what's really fascinating is that there are experiments done by this gentleman over here, Hans Spemann, where he took specific cells of an early embryo and he grafted them to new positions on another embryo to basically show the role of morphogenesis. So we're really talking about this term of embryonic induction. And what embryonic induction is that you have a group of cells that ultimately induces or directs the development of a second group of cells, which in a way is quite fascinating. You have cell-cell communication. So if you plant the ectoderm in a new location, you can ultimately get gene expression to become changed. So in terms of the first step over here, the first factor, if you may, is that location is extremely important. So I think that makes a lot of sense. The next kind of factor that we're really looking for here is cytoplasmic segregation. So cytoplasmic segregation, or unequal cytokinesis, factors unequally distributed cytoplasm. So factors are unequally distributed in cytoplasm. There we go. That's what we're going for. So unequal cytokinesis is when you have specific factors that are found within the original parent cell that are going to be unequally distributed in the daughter cell. So if we can see over here, the white factors here become part of one daughter cell, but it's not really not part of the other daughter cell, and that ultimately can have variation in the cellular fate. So cytoplasmic determinants are unequally distributed by often the cytoskeleton, and therefore cytokinesis can ultimately lead to different daughter cells. So again, cytoplasmic segregation or unequal cytokinesis, you have factors that are unequally distributed in the cytoplasm, and they may end up in daughter cells they may end up in some daughter cells, but not others. This is not really worded well. I need to change that. Okay, but let's move on. So the next step, of course, is we're looking at cytoplasmic segregation. What about within it? Well, each individual cell has variation in polarity. So there's a top and a bottom of the cell. We often call it the animal and the vegetal poles. And ultimately, when we're looking at situations of cellular division, we can also think about how there can be variation within certain concentrations of nutrients or signal molecules. So for instance, a calcium can have high influx rates upon sperm entry. Whereas if you look at situations like this, you can see that there's variation in the types of molecules that are found in certain regions of the cells, which in many ways, if you think about it, through cytoplasmic segregation can lead to unequal distribution of various factors, which again comes back to this thing over here. Cool. Okay. So factor number 3 is this thing that we just talked about, which is induction. So induction is just cell-to-cell communication. You have a factor that's secreted by a certain cell that induces other cells to differentiate. So the closer a cell is to an inducer, that particular signal, the more it can be activated/inactivated through signal transduction cascades. So an example is that vulva formation in roundworms is mediated by HGF over here, the epidermal growth factor, and then vertebrate limb formation is often mediated by sonic hedgehog genes secreted by cells of the zone of polarizing activity, or ZPA. Lots of information over here, but really the key idea is that cells in a developing embryo influence one another's developmental fate via chemical signals through this process called induction. Okay, so this is a topic that I guess a lot of people already have an idea on, so I'll go over it briefly, and that is cell death. So cellular death is important, and it's broken down into sort of two main groups. We have apoptosis, which is programmed cell death, which is where cells must be removed during morphogenesis and development, programmed by the individual body. And then, of course, we have necrosis, which is often damage to the cell themselves. And in necrosis, we're often talking about situations such as oxygen or nutrient limitations, which can lead to swelling and bursting of the cells. And the release of these contents, so when a cell bursts and you have prostaglandins and potassium that's being thrown out into the extracellular space, that can lead to inflammation. So it can potentially have a negative effect on the body, which is why here the key idea is that apoptosis may be a more preferable form of cell death, especially in development, because you don't want that inflammation to be going on around your body. Now the question, of course, is why would apoptosis occur? What's the benefit of having programmed cell death? Well, one of them is because the cell is just no longer needed. So if you have connective tissue like webbing between our fingers on a fetus, that's not necessarily needed once the baby is born, so we don't really need any webbing between our fingers, and so apoptosis gets rid of that connective tissue, and that's all great. So that's one reason. Another reason is simply the fact that you have old cells that can become, prone to mutation and cancer, so you got to get rid of those and try to destroy them before they start replicating out of control. You can just have cells that outlive themselves, so you have blood cells, epithelial cells that die after certain number of days or weeks just simply due to natural damage. And then finally, last but not least, you have a great turnover of certain neurons within the brain during development, and that can also be mediated by apoptosis. So once again, apoptosis may be a preferable form of cell death, especially in development, because you avoid inflammation and other problems that can occur within necrosis and cell death because of certain damages. Okay? Awesome. So what exactly causes potentially apoptosis to occur? Well, there are many factors, but a few brief ones here. First of all, you have lack of mitotic growth, so you don't have lack of growth factors that can contribute to apoptosis. You can have recognition of damaged DNA, so if the DNA damage is just way too high, there's nothing you can do about it, then you might just go ahead and destroy the cell to prevent it from staying around and replicating. And within the process of apoptosis, you actually can have these enzymes called caspases and other secondary enzymes there that can ultimately cause this cascade that will break down proteins, break down cellular membranes, and then that ultimately leads to the destruction of the cell. And so there we have it, apoptosis in a nutshell. Okay, so I decided to throw this section over here. It really probably better fits into the fertilization and development part, but I think that it also has some impacts into the whole concept and discussion of cell fate, so I decided to put it here instead. So after gastrulation, remember formation of our germ layers within the gastrula. What happens? Well, we can have segmentation along the anterior and posterior axes, and that's very common in organisms like insects. And then after that, we have organogenesis. Now, organogenesis is very important because we can have neurulation, which is the formation of the neural tube along the posterior and anterior axis, and that eventually develops into our nervous system. We can also have muscle development, which occurs after neurulation from these things called the somites. If you take a neuroscience course, you're going to go into a lot more detail on this, I can tell you from experience. But for now, I think this is more or less sufficient. But keep an eye on your notes and see what your professor has to offer in terms of what you need to know for the exam. Okay, what's next? Okay. So segmentation. So how is segmentation itself determined? Well, there are genes that contribute to it. So we have gap genes that organize broad areas. We have pair rule genes that divide the embryo into units. We have segment polarity genes that determine boundaries and the anterior, posterior organization of individual segments. So a lot of genes can help contribute to segmentation. And obviously, when we look at the distribution and development of certain critical sections within an organism, we have to talk about Hox genes. So Hox genes or homeobox genes are very important because they're expressed in different combinations along the length of the embryo, and they ultimately determine what each segment will ultimately become. So is it going to be the head? Is it going to be the thorax? Is it going to be a section of the abdomen? And it's very funny because in many ways, Hox genes are very conserved between many animal lineages. But we realize of the importance of Hox genes when we look at, for instance, mutants. So there's an antennapedia mutation where you can get legs to grow in place of the antenna in fruit flies. And same thing in fruit flies, you can have bithorax mutations, which basically can cause the fruit flies to grow an extra pair of wings. So that's in a way pretty cool. And it's all about Hox genes. And this is more of an evo-devo topic, but some brief information on it is usually important. But it might be good to potentially look at something like this slide to kind of get the basic gist of it. So you have maternal effects genes that determine the anterior-posterior axis and induce the 3 classes of segmentation genes. We then have your gap genes, which just kind of define general broad areas. And they regulate these pair rule genes, which kind of refine the segment locations and ultimately regulate the processes a little bit. And then we have segment polarity genes, which determine the boundaries and anterior-posterior orientation of each particular segment. And then finally, we have Hox genes, which define the role of each segment. Again, whether it's going to become the head, whether it's going to become the legs, or whether it's going to become the wings or the antenna. And that can have, obviously, extremely important effects on the development of the organism. And it's also going to tie itself into crucial parts in relation to things such as cellular fate. So that ultimately kind of sums up the whole concept of segmentation, as well as ideas relating to final steps in organ and system development. So I hope you found that at least mildly useful, and as always, best of luck studying, and good luck on the exam, everyone.